Hello everyone, welcome back to Magic School, and we're moving forward in our toxicology course. Now we're going into genetic toxicity to see how these toxicants actually affect our genes or actually affect our DNA, and then cause about mutation or cause about changes in our genetic structure, changes in our genotype. So yeah, let's get to that. So now, like, damage to our genetic material involved in cell division is caused by chemicals. As a result of interaction of chemicals with our DNA, they could be damaged or they could be changed to the DNA, thereby causing this DNA, when they are involved in cell division, could cause mutation and all that. The degree of effect of such changes depend on the extent to which the change occurs. Like it could probably just be maybe a point mutation, or it could even be an entire codon that is being changed, or an entire codon that is being deleted, or it could be a frame shift mutation, which are things we're eventually going to. So these changes could occur in a region of DNA where there are no critical codes, or the change may be of no significance. Now, there are some places on the DNA strands that when changes occur, it could actually be lethal. It could actually be lethal. It could actually be of no significance, having no effect at all. It could actually be beneficial. It could actually be beneficial to the organism. Now, once the DNA, or once these chemicals have access to the nucleus, they can be able to interact with the DNA in a variety of ways. So once these substance or chemicals are able to get into the nucleus, interact with the DNA, it can thereby cause change and chemical changes in the DNA. Now, these antibiotics can be incorporated or directly react with the DNA, and they do this by direct interaction with the DNA damage, base substitution, and base addition and deletion. But for this video, we'll be talking about just the first two: direct interaction with DNA and DNA damage. So yeah, let's get into it. So now, direct interaction with the DNA. Now, the first thing to consider here is the alkylation of bases. These are the nitrogenous bases. Alkylation of these nitrogenous bases. So now, these alkylating agents, they are seen to react with electron-rich atoms. So now, in these nitrogenous base, the electron-rich atoms you have are the nitrogen and the oxygen atoms. So now, these alkylating agents are seen to actually react very well with the nitrogen and oxygen. Now, there are some nitrogenous bases that have actually vulnerable sites for these alkylating agents. Now, for guanine is N7 and O6. For adenine is N3 and N7. Now, a quick way to remember it is, if you look at something, adenine and guanine are actually purines. Now, we now have an example of an alkylating agent, vernichloride. Now, vernichloride is seen to react actively with N1 of adenine, N3 of cytosine, and N7 of guanine. Now, a simple way to also remember this, which is quite how I learned it, is like adenine is 1, like in the alphabetical order, A1. So N1, the first nitrogen. Now, for cytosine, ABC, that is the third one. So the third nitrogen, N3. Now, guanine, when you count the alphabet, guanine is G is the 7th alphabet, so N7. So that is just the way to put it. And then you notice that adenine and guanine are purines, and cytosine is a pyrimidine. So just to get that. And then we have also alkylnitrosamine. Now, this reacts with O6 and N7 of guanine, as I mentioned, which are actually vulnerable sites for alkylating agents for guanine. So now, the formation of these adducts will in some cases affect the ability of the base to form nitrogen bonds across the DNA double helix. Now, the alkylating agents covalently bind to the nitrogenous bases. The adducts are formed. Formation of adducts have been formed. Then now, it impairs the formation of hydrogen bond in double helix. Now, we know that hydrogen bond between the nitrogenous bases is what holds the two strands together. Now, there's impaired formation of this hydrogen bond, and now this causes problem when there's replication, and then pairing causes problem with replication, and then pairing later on, and then base substitution. The particular sites and type of base alteration depends on the particular reactive metabolite. Now, formation of adult can be regarded as structural modification of DNA. A change from the normal of how it was supposed to be could eventually cause a mutation. And now when they are bulky adults, these bulky adults, which are like large adults, can cause misreading of the code, wrong interpretation of the code during translation or replication, and it can affect the polymerase action. That is RNA polymerase, the DNA polymerase. It can actually affect their action, and then it can also stimulate DNA repair. Now we are talking about bulky adults, can actually stimulate DNA repair, and then this DNA repair may eventually be prone to error, and then that could cause a mutation and all that. If an alteration is not fixed when the DNA is undergoing repair, or when it's actually being replicated, and then wrong pairing occurs, the nitrogenous base pairing is changed, this could eventually be a mutation. Now the mutation will be passed on to the daughter cells after cell division, and the error will become fixed. It won't be able to be changed again, and then it goes on and on and on, and that's how we get mutations. Now, so adults are actually quickly repaired, some are actually quickly repaired, and some are unstable, and then some cause mutations and some actually do not. Some actually cause mutations in the genotype, but now we don't really see it in the phenotype, probably because it's affected in a place of where there's no critical code or critical genes. So it's not really, yes, the genotype is changed, but the phenotype is not expressing that change. DNA damage. Now this DNA damage, it could also be as a result of the interaction of these chemicals and alkylating agents. Now the first thing to look at here is apurinic sites and apyrimidinic sites. What we'll be considering here is guanine. Now when the N7 the nitrogen atom is alkylated by an alkylating agent, the glycosidic bond between the sugar and the nitrogenous base become quite unstable, and then the purine, which is guanine, is eventually displaced, causing an apurinic site. Thus, these apurinic sites are formed. Insertion of an incorrect base at the PM can then cause a mutation. Now we also have cross-links and pyrimidine dimer in a strand. DNA has two strands. This pyrimidine dimer is a pair of pyrimidine bases within a nucleic acid covalently linked by a cyclobutane ring. So now the DNA sequence was GC80. Now C and T interact, forming a cyclobutane ring that holds them together, and then this causes misreading or incorrect functioning of the DNA at the end. So that is what this pyrimidine diamond is about. Now intercalation, this is more like an insertion. Intercalation is more like an insertion into elements or into a layer, like let's say there's ABC layer, and then something is being inserted between A and B. So that's what I mean. Intercalation by planar molecules and cancer drugs and alter the structure and function and lead to mutation by base insertions. Now the first molecule we have here, which is a planar molecule, is acridine, and then the next one is vinblastine. So a simple way to remember the last two, which are actually cancer drugs, vinblastine, just remove the V and the E at both ends, we get vinblastine, increastin, and then just add the V and E to it at the back, vinblastine, vincristine. Actually, when you put things in syllables, you actually tend to get it better. So yeah, that is that. Now we can also have oxidative damage to the nitrogenous bases. We have the reactive oxygen species, and then some of them are actually produced in the body. Now it can convert, you can see damages or changes from cytosine to uracil, or from guanine to 8-oxo deoxyguanosine. So it can actually be converted to that. Now this 8-oxo deoxyguanine is seen to pair erroneously with adenosine. Now when this happens, this causes base pair substitution or base pair change, and then this causes a whole lot of other problems later on that we'll be explaining in the next video. So the endogenous RNA is seen to cause more damage to the DNA than exogenous mutagens. So now the mitochondrial peroxisomes is where this RNA is actually being produced, and then these RNAs, if they are not properly metabolized, or if they are not properly detoxified, it can interact with the DNA, both the mitochondrial DNA and the nuclear DNA. But now this mitochondrial DNA are seen to be more exposed to these RNAs, or more easily affected by these RNAs, because now this mitochondrial DNA, they lack histones. they don't have histones surrounding them, and also they are in close proximity to the site of production. Now, I said not too long that this ROS actually being produced by the mitochondria, by the normal metabolism is produced by the mitochondria, and then we have mitochondrial DNA, so they can easily interact with this DNA when they are not easily removed from the body. The last thing to note here is inflammation. Now, inflammation can also be responsible for oxidative damage of DNA via inflammatory cells such as the phagocytic cells. An example we have is the neutrophils. So now these neutrophils, they act to this is the cause, something we should look under immunology later on. But now these neutrophils they produce oxidizing agents that to actually act on this foreign substance that come into the body. An example of this substance we have are actually hypochlorite, superoxide, nitric oxide, and hydrogen peroxide. So now all these they have to act on antigens to kill antigens or to remove antigens from the body so they don't cause effect on our body. But now these oxidizing agents that I just mentioned, they can actually interact with the DNA based. It is seen that in tissues, in inflamed tissue, there is more of 8-oxodeoxyguanosine, which is actually a product of oxidative damage than in normal tissue. That means inflamed tissue has more of what I just mentioned than the normal tissue. Yeah, so that is all for today. So thank you for listening. So that is all for today. That's all we'll be talking about for today. But in the next video, we'll be going into how this actually affects the DNA, causing base substitution, deletion, insertion, and all that. So yeah, let's look forward to that. And don't forget to like and subscribe. Thank you.